Good afternoon, and welcome back to AP Biology Semester 2. We're with the big idea number 2, free energy. And we are still in the 6th chapter, the tour of the cell. Today we are going to talk about mitochondria and chloroplasts, how they change energy from one form to another. Organisms transform the energy they acquire from their surroundings. In eukaryotic cells, mitochondria and chloroplasts are the organelles that convert energy to forms that cells can use for work. Mitochondria, the singular form is mitochondrion, are the sites of cellular respiration, the metabolic process that generates ATP by extracting energy from sugars, fats, and other fuels with the help of oxygen. Chloroplasts, found in plants and algae, are the sites of photosynthesis. They convert solar energy to chemical energy by absorbing sunlight and using it to drive the synthesis of organic compounds such as sugars from the carbon dioxide and water. In this section, we will also consider the peroxisome, an oxidative organelle that is not part of the endomembrane system. Like mitochondria and chloroplasts, the peroxisome imports its proteins primarily from the cytosol. Okay, now mitochondria is a vessel for chemical energy conversion. Let's talk about that for a minute. Mitochondria are found in nearly all eukaryotic cells, including those of plants, animals, fungi, and most protists. Even in exceptions such as the human intestinal parasite Giardia, and some other protists. Recent studies have identified closely related organelles that probably evolved from the mitochondria. Some cells have a single large mitochondrion, but more often a cell has hundreds or even thousands of mitochondria. The number correlates with the cell's level of metabolic activity. For example, motile or contractile cells have proportionately more mitochondria per volume than less active cells. Mitochondria are about 1 to 10 micrometers long. Time-lapse films of living cells reveal mitochondria moving around, changing their shapes and fusing or dividing in two, unlike the static cylinders seen in electron micrographs of dead cells. The mitochondrion is enclosed by two membranes, each a phospholipid bilayer with a unique collection of embedded proteins. The outer membrane is smooth, but the inner membrane is convoluted with enfoldings called cristae. The inner membrane divides the mitochondrion into two internal compartments. The first is the intermembrane space, the narrow region between the inner and outer membranes. The second compartment, the mitochondrial matrix, is enclosed by the inner membrane. The matrix contains many different enzymes as well as the mitochondrial DNA and ribosomes. Enzymes in the matrix catalyze some steps of cellular respiration. Other proteins that function in respiration, including the enzyme that makes ATP, are built into the inner membrane. As highly folded surfaces, the cristae give the inner mitochondrial membrane a large surface area, thus enhancing the productivity of cellular respiration. This is another example of structure-fitting function. All right, now let's talk about chloroplasts. The chloroplasts, they capture light energy. Okay, the chloroplast is a specialized member of a family of closely related plants organelles called plastids. Some others are amyloplasts, colorless plastids, that store starch, amylose, particularly in roots and tubers, and chloroplasts, which have pigments that give fruits and flowers their orange and yellow hues. Chloroplasts contain the green pigment chlorophyll, along with enzymes and other molecules that function in the photosynthetic production of sugar. These lens-shaped organelles, measuring about 2 micrometers by 5 micrometers, are found in leaves and other green organs of plants and in algae. The contents of a chloroplast are partitioned from the cytosol by an envelope consisting of 2 membranes separated by a very narrow intermembrane space. Inside the chloroplast is another membranous system in the form of flattened interconnected sacs called thylakoids. In some regions, thylakoids are stacked like poker chips. Each stack is called a granum, plural form, grana. The fluid outside the thylakoids is the stroma, which contains the chloroplast DNA and ribosomes as well as many enzymes. The membranes of the chloroplast divide the chloroplast space into 3 compartments: the intermembrane space, the stroma, and the thylakoid space. In Chapter 10, you will learn how this compartmental organization enables the chloroplast to convert light energy to chemical energy during photosynthesis. Okay, now that we've talked about the mitochondria and the chloroplast, let's move on to the peroxisomes and its oxidation. The peroxisome is a specialized metabolic compartment that is bounded by a single membrane. Peroxisomes contain enzymes that transfer hydrogen from various substrates to oxygen, producing hydrogen peroxide as a byproduct, from which the organelle derives its name. These reactions may have many different functions. Some peroxisomes use oxygen to break fatty acids down into smaller molecules that can then be transported to mitochondria, where they are used as fuel for cellular respiration. Peroxisomes in the liver detoxify alcohol and other harmful compounds by transferring hydrogen from the poisons to oxygen. The H2O2, which is hydrogen peroxide, formed by peroxisomes, is itself toxic. But the organelle also contains an enzyme that converts hydrogen peroxide to water. This is an excellent example of how the cell's compartmental structure is crucial to its functions. The enzymes that produce hydrogen peroxide and those that dispose of this toxic compound are sequestered in the same space, away from other cellular components that could otherwise be damaged. Specialized peroxisomes called glyoxysomes are found in the fat-storing tissues of plant seeds. These organelles contain enzymes that initiate the conversion of fatty acids to sugar, which the emerging seeding uses as a source of energy and carbon until it can produce its own sugar by photosynthesis. Unlike lysosomes, peroxisomes do not bud from the endomembrane system. They grow larger by incorporating proteins made primarily in the cytosol, lipids made in the ER, and lipids synthesized within the peroxisome itself. Peroxisomes may increase in number by splitting in two when they reach a certain size. And we are going to stop here for today. And the next time we talk, we will be talking about the cytoskeleton and its network of fibers that organize the structures and activities in the cell. Until next time, ta-ta!